All right. Well, thank you for that introduction and for the invitation. Et s'il y a des questions que vous voulez poser en français, je suis toujours capable de répondre en anglais. So, where am I? Where am I without my glasses? Blind is where I am. So, yeah, the kind of unifying thing in my career, I guess, was positron emission tomography, which is a technique for imaging in the brain of substances. It doesn't matter what substance. If you can make it radioactive in the right way, you can see where it goes. So pharmacologists like it because you can engineer biomolecules that are radioactive in the way that can be detected by PET. And so I'm going to talk a little bit about how some efforts towards using the PET technology and allied methods to get a better understanding of this interesting molecule DMT and N-dimethyltryptamine with respect to its pharmacology and metabolism. So, as you know, I'm sure you all appreciate that the ayahuasca is the term to describe a traditional usage of a mixture of usually two or more plant stuffs that are native to Amazonia and Central America, one of which contains DMT, dimethyltryptamine, which is a potent hallucinogen, and the other containing monoamine oxidase inhibitor, usually harmine, and similar molecules. And the interesting thing about DMT is that it's nearly inactive orally. As far as we know, you can probably eat it by the spoon. You could put it on your cereal. It wouldn't do anything because as soon as it enters the gut, it gets broken down by monoamine oxidase and never gets a chance to obtain sufficient concentrations in the blood to do anything. Intravenously, it's extremely potent, and it can also be insufflated and smoked and so on. But the usual approach for administration is in the traditional context of these brews, such as you see here, which contain the one plant Banisteriopsis that contains the MAO inhibitor and something like, what's it called, the psychotria or other sources of DMT. Also mimosa is another popular source for DMT. And taken together, it produces this profound experience with visuals and insight and the whole gamut of a psychedelic experience. Now, going from the jungle to the hard lab, I first got interested in this some decades ago when I became aware of PET research showing that tobacco smoke has an MAO inhibitor in it. And this was discovered by colleagues, I guess I could say, at the Brookhaven National Laboratory in the United States a long time ago, like 40 years ago. They were doing initial experiments. Can you see the cursor here? Where they had produced a radioactive molecule called carbon-11 deprenyl, but never mind that. All you need to know is that it's an MAO ligand. And then they injected it intravenously. It's radioactive and can be detected by the PET scanner. And then they produced this kind of figure showing the distribution of that tracer in a healthy person. And you can see that the brain lights up because there is a lot of that kind of MAO in the brain. It lights up the myocardium, the heart, because the same enzyme is present in the sympathetic nerves that innervate the heart. And it was deposited heavily in the kidneys and liver. However, one of their volunteers, unhypothesized, there was no prior expectation here. He was just an historically early volunteer in this kind of research, proved to have almost no MAO. And why was that? There is a rare mutation, some Dutch family that lacks MAOA, which is not very good behaviorally. But in this case, they determined that he was a smoker, a heavy smoker. And in fact, something in the tobacco smoke, specifically in the pyrolysis of tobacco, is inhibiting MAO. And there's some reason to believe that it may be harmine itself, the same substance present in ayahuasca. So the Brookhaven work from I forget when it was published, like almost 40 years ago, in the early phase of this kind of endeavor of molecular brain imaging, inspired my interest in ayahuasca. The whole concept that the endogenous monoamine oxidase substrate could include dopamine, which is metabolized by both forms of MAO, or serotonin, which is present in the brain and in the blood, and is exclusively metabolized by MAOA, which is the same enzyme involved in ayahuasca. And does the MAO inhibition from tobacco smoke alter dopamine serotonin metabolism in brain in smokers, potentially altering the effects of nicotine itself? So that was my thought experiment, was that tobacco smoking was ayahuasca light. That is a generalization kind of the same principle of ayahuasca, where an MAO inhibitor alters the metabolism of another substance. And I did some experiments in that direction, but I wasn't able to prove it. I still think there's something there because, as I was just saying, it could explain possibly the failure of nicotine replacement therapy in tobacco dependence, because maybe having the nicotine is not enough. Maybe you need to have the nicotine and MAO inhibition. And as far as I know, no one has tested that. I guess I should patent it. So, since coming to Berne, in the context of a successful grant proposal, actually 2 or 3 years ago now, and funded by the SNF, I wanted to investigate some very basic aspects of the pharmacology of DMT in conjunction with the MAO inhibitor harmine. And this this is in association with Professor Scheidecker from Uni Zürich, and our mutual student Clemens Egger, and Elena Eicher is probably known to you. Recently, just weeks ago, this big massive review came out, which is kind of nice. I think it's there have been a number of such reviews, but this is the most recent and comprehensive one that I'm aware of. To just review this whole topic of the interaction between monoamine oxidase inhibitors and DMT and other tryptamines, which are also metabolized by MAO. So I think it's a pretty good reference if you need to review that literature. And taken from that figure from that same paper, we see some of the relevant molecules, such as tryptamine, 5-MeO-DMT, DMT itself, and we produce this figure just to show that they have a structural motif of the indole amine structure, which is shared with LSD, serotonin in the brain, and a lot of other things. And the other class of molecules present in the ayahuasca mix is the so-called beta-carbolines, which, if you look closely, they are in fact cyclized tryptamines. So it all comes down to tryptophan and its various metabolic pathways. And whereas these substances are psychoactive in varying degrees, the lower figure is these compounds have little psychoactivity. That's still debated a little bit. Harmine and beta-carbolines may have some intrinsic psychoactive properties, but weak. However, they are potentiators of the action of other active substances that would otherwise be metabolized by monoamine oxidase. This is all very familiar. And in that same paper, Clemens produced a nice figure showing the metabolic pathways where the beta-carbolines are not inert, but they are metabolized by various routes, but they have in common the ability, here harmine, to block the metabolism of DMT to its inactive metabolite, indole acetic acid. The red arrows indicate inhibitory function, and it's mechanistically just a simple competitive inhibition, where the beta-carboline sits on the active site of the enzyme, monoamine oxidase A enzyme, and blocks it. And because it's a reversible competition, it's very difficult to get complete blockade, because it's this kind of S-shaped function. The more you have to add more and more of the harmine to inhibit completely the enzyme monoamine oxidase. So, in there's general agreement that the key target of most psychedelic substances, unless you include ketamine is in that category, but the classical psychedelics all seem to act at serotonin receptors in brain and in the periphery. A specific class of serotonin receptors, 5-HT2A sites, which are the common target of most classical psychedelics, but not the exclusive target. So, for example, LSD binds to these receptors and a whole lot of others. It's one of the more non-selective drugs you could imagine. However, in the main, the experiential aspect of LSD effects, as far as I know, is mostly obtained by activating the serotonin 2A receptors. The other receptors may add depth and dimension to the experience, but the essential part of it is probably 5-HT2A. How do we know this? Well, because ketanserin, which is an antagonist of serotonin 2A receptors, rescues most of the psychotropic effect, which is to say, it's a trip killer, you could say. And ketanserin is fairly selective for 2A receptors. So if you block the 2A receptors, then classical psychedelics like LSD and psilocybin no longer work. The case is less clear for DMT. The only experiment of which I'm aware where they attempted to do this was a naturalistic ayahuasca setting in South America where researchers at Sao Paulo, I think, administered ketanserin to ayahuasca participants and monitored their reactions. And there was a substantial reduction, but incomplete reduction. So in other words, ketanserin at the dose they gave, didn't completely block the experience. And that's interesting because it suggests there might be other pharmacological components than the classical one. So how do we get at this? Many different techniques and procedures could be used to investigate this in the lab. But the one that I've been most involved with is positron emission tomography. You already saw an image showing the MAO in brain of smokers. But the principle is that you wear a hat. Actually, modern PET scanners, you lie down, but in the very first PET scanner, I think from the 1960s in this figure, was a ring of detectors. And the principle is that a radioactive molecule, once it finds its way into the brain, if it's endowed with a particular type of radioactivity, will produce, I can't see, oh yeah, you can see the red star? Yeah. An emission of 2 photons at 180 degrees due to the particular mode of decay and annihilation of the positron. And it's summed up by this figure, where on the left-hand side you see the positron, which has somehow found its way into the brain, being carried by the radiopharmaceutical that decays to release a positron, hence positron emission. The positron itself you can't see, but it wanders around in the brain for some time, not a long time, less than a nanosecond, before it encounters an electron, which your brain is full of. And when the two meet, it's the fatal attraction of the positron-electron pair, where they form a very short-lived molecule, a kind of atom called positronium, that for reasons that physicists could explain is unstable, and immediately the positron-electron pair annihilate each other, and all the energy from E equals mc squared that was bound up in that electron-positron pair is released all at once in 2 photons at 180 degrees, one going up and one going down. So if you count millions of those events in the scanner, you can produce what might be called a source map. You can see what the radioactivity concentration was as a function of time after the injection of the radiopharmaceutical. And the particular radiopharmaceutical is a matter for radiochemistry, where skilled chemists are able to integrate these very short-lived positron-emitting radionuclides, in this case fluorine-18, into a tracer molecule, in this case basically altanserin, almost the same as ketanserin, slightly different structure. And that requires some fast work because fluorine-18 differs from the abundant natural fluorine-19, which is completely stable until the end of time, as far as we know. But fluorine-18, being neutron deficient, really doesn't want to stick around in this universe very long. So it falls apart within a matter of hours by this mode of decay that releases a positron. The positron then meets an electron in the brain and releases these two photons. And so this is an image produced by my colleague in Vienna showing the, the distribution of altancerin, fluorine 18 altancerin binding sites in a series of humans. They were actually averaged. So this is several brains stacked on top of each other. And you can see certain features that it's highly abundant in the cerebral cortex, especially the frontal and paralimbic cortex, or orbitofrontal cortex, and in the occipital, you could say, visual cortex. I wonder if that has something to do with the phenomenology of psychedelics. And notably, it's a complete void in other structures. You see, these are subcortical structures of the brainstem, the cerebellum. So it's really a cortical neurotransmitter receptor. And this is where presumably psychedelics are acting, at these receptors. So serotonin 2A receptors have almost an exclusive distribution in the cerebral cortex, notably in the visual cortex and the frontal cortex. Now, stepping back away from that for a moment, I'm just going to quickly go over some of the work that I did decades ago in Denmark, when I first became interested in this topic. And it became apparent to me that this beta-carboline here, harmol, could be labeled with carbon-11, which is another positron-emitting, very short-lived radionuclide. It has to be cooked up fresh and used within a matter of 90 minutes, because by the end of 2 or 3 hours, it's all gone. So they're able to manufacture, using a cyclotron, this carbon-11, and use some quick chemistry to integrate it into the precursor molecule to produce a new molecule, harmine, which is labeled with carbon-11. And so now we have an MAOA ligand. And we did studies in the pig, which is a sort of a compact model of the human brain. And you see the ghost of the structural MR underneath. And on top, you see the distribution of harmine binding. And it's clear that it's very abundant. In the lower figure, the same pigs were scanned after blocking with a blocker of MAOA that completely negated the specific binding. But the specific binding is not homogeneously distributed in the brain of pigs or humans, but it's present ubiquitously in the cerebral cortex, but it's also present in the subcortical structures, which include, I think this is the amygdala, the dorsal thalamus, and the brainstem, and for those who are interested in the locus coeruleus. And so it's not uniformly abundant, but it's very interesting, I think, that we know from other lines of work that these are the serotonin brain regions. So MAOA has a close affinity for serotonin in the brain, which is most abundant in exactly these structures. So why? Because MAOA is the enzyme for metabolizing serotonin that's formed in the brain. So it's not coincidence that there's some kind of overlap. However, it's not a complete match with the serotonin 2A receptors, which are more exclusively found in the cerebral cortex. So that's where harmine sticks in the brain. DMT, dimethyltryptamine, that has survived first-pass metabolism, meaning after oral administration in a concoction of ayahuasca, It runs the gamut of the gastrointestinal system, which has a lot of MAOA in it, which is why there's extensive first-pass metabolism, and such that DMT is practically ineffective given orally unless you pre-block the MAOA. But MAOA is also highly abundant in the brain, as you can see in this figure. So some of the DMT, even if it's gotten into the brain, it's exposed to the MAOA that's in the brain, not in the gut. So brain MAOA activity ultimately limits the duration of action of DMT also. Is that clear to you that, what the DMT, first it has to pass through the gut, and runs the risk of being inactivated by MAO. But then it enters into circulation through the liver and into the general circulation, and then some of it gets into the brain. And there it doesn't stay forever because it's also being inactivated by MAO in the brain. So, you know this toad, I guess. It's one of nature's wonders, the Sonoran toad, which contains, shall we say, venomous or kind of glands on its back. Not like the cane toad in Australia, which is just simply poisonous. This poor toad is apparently being hunted to extinction because people think that it's very important to smoke them. And it contains bufotine, which is essentially DMT. Yeah, I mean, it's the same molecule except it has a hydroxyl group. And then, as you know, you've also heard of meo-DMT, which is the same molecule except it has a methyl group here. These additions don't change the basic pharmacology much. It's still have in common the ability to produce psychedelic experiences. The main thing is that you have to have this N-dialkyl group. It isn't even essential that it's dimethyl, it could be diethyl or dipropyl. So again, back in Denmark, thinking about this in a discussion with the radiochemists there. Radiochemists are the people who have the skill set to make these radioactive molecules. We realized that we could take this precursor compound, which I had to buy on the black market at that time, and using certain chemical procedures, integrate the carbon-11 into it. So I thought it was a cool idea. Let's find out where meo-DMT sticks in the brain, yeah? Should be interesting. So in order to test that hypothesis, that procedure, once the chemists had established the method for making this meo-DMT that's radioactive, we obtained dynamic PET recordings. That means you inject the tracer and then take a series of images of the emissions from the brain to get the kinetics as it enters the brain and sticks. And in some pigs, we co-administered other drugs to see if we could block or displace the expected binding of the meo-DMT in the brain of these living pigs. They're unconscious, by the way, because they're anesthetized, so they don't experience anything as far as we know. And the net result is here. These figures are the averages of 3 PET scans in pigs where they were scanned with 5 carbon-11 meo-DMT, and the binding was calculated by some means, and you can see it's exactly not where the serotonin 2A receptors are. If you recall, they were found in the cerebral cortex. To make matters worse, treating the pigs with ketanserin, the 5-HT2A antagonist, had no effect on the binding. So we can say with absolute certainty that meo-DMT doesn't stick to serotonin receptors at the dose that we gave it, which is a microdose. It's sticking to something else, though. It just wasn't displaceable by ketanserin. And the distribution here, do you know, most of you, some basic neuroanatomy? Well, this is the ventral striatum. This is the nucleus accumbens, which is the site of the reinforcing aspects of cocaine and psychostimulants. And more significantly, it's in the ventral striatum. You can't really see it here because here's this is the ventral striatum. Is the lower you get in the brain of the pig, this is the ventral surface, the belly, so to say, of the pig, the more intense the binding. And it reminded me a lot of the known distribution of a certain kind of dopamine receptor in the brain. Unfortunately, I had to abandon that project, and so I was never able to show specifically my conjecture that meo-DMT is first sticking to dopamine receptors. How would I have done that? Well, I would have had to pre-treat the pigs with a selective dopamine blocker. But it certainly looks familiar. You have to agree that this D3 ligand is binding in slices of rat brain in the ventral striatum, which is really the same corresponding region as in the ventral hotspot in this binding. So I never managed, you don't have to feel sorry for me, but I never managed to get that study published. I think we submitted it 6 or 7 times back in the day, and people just said, this is rubbish, it's a negative result, not worth publishing. I guess I should try again, but I'm still kind of PTSD about this paper. So nobody likes a negative result. I mean, this is, unless it's a COVID test there. So I guess that was my if you could take it as a kind of encouragement, if you are getting to a point in your studies where you're publishing or trying to publish papers, reviewers can be wrong, and the bias of the times, that research was done before the renaissance of psychedelic medicine. I think I did it 20 years ago, and people just weren't interested. And so I guess maybe I should try to find the courage to resubmit it this time, because even a negative result is sort of interesting, because what do we see? Two things. DMT is not significantly binding at this dose to serotonin receptors, and second, it seems to be sticking to a dopamine part of the brain, and that should be interesting, even if I can't completely explain it. The most widely used PET tracer is not these exotic pharmaceuticals that I've been talking about, but radioactive glucose. Glucose on the left-hand side, and its partner fluorodeoxyglucose, labeled with this positron-emitting hot fluorine. And it's universally used in nuclear medicine departments around the world because it's so darn useful for detecting tumors. Because most solid tumors are avid, have a high metabolic rate, and are greedy for glucose. And they can't tell the difference between glucose and fluorodeoxyglucose. So you can see this unfortunate person has a tumor of it's the pulmonary vein there, and certain metastases elsewhere. But they weren't particularly interested in what, for me, the interesting part is that it had extremely high uptake in healthy brain, because this person's brain was not affected by the disease. And yet nervous tissue, neurons and brain tissue, are as hungry for glucose as any tumor. And that's just the property of neurons. It's expensive to maintain neurons in their healthy polarized state. It consumes a lot of energy. Weight by weight, you've done these calculations. I think the brain is using about 20% of your energy budget, even if you're sleeping all the time. It goes down a little bit when you sleep. I don't see anyone sleep here, but that's good. The fluorodeoxyglucose can be allowed, it serves its main purpose these days in clinical oncology for diagnosing certain kinds of tumors, but its principal history was in brain research, because the brain is so avid for glucose, it's very useful to use in challenge conditions to see if drug A or drug B alters the energy consumption of the brain. And here we refer back to pioneering work by Franz Vollenweider, I guess a name familiar to you, in Zurich, who was really on this before it was fashionable. He published this paper in 1909, I think it was, so well on 25 years ago, where they gave fluorodeoxyglucose to healthy volunteers in two conditions, at baseline and again after consuming psilocybin at a psychoactive dose. This figure has nothing to do with the paper. It's just an example of what fluorodeoxyglucose uptake looks like in brain. You see that it's mostly in the cerebral cortex, and it's highly elevated in the occipital cortex, and etc. So you can quantify this. You can turn these PET images into a quantity which corresponds to the rate of glucose consumption. And he did exactly that with his group in the two conditions and compared before and after. They present boring bar graphs because 25 years ago there was no technology for producing these maps. It didn't exist yet, these averaging. But so each brain had to be considered by itself, and you do a little circle around the occipital cortex, the frontal cortex, etc., and evaluated the uptake. And what they saw was across the board a significant and substantial increase in glucose consumption between the baseline and the psilocybin conditions, on average about 15%. And this is extraordinary because that's a big increase in glucose metabolism. Most drugs no other drug that I'm aware of, other than something that causes convulsions, would do this. Nobody has been able to reject this finding. They saw what they saw. There's nothing wrong with the design. I've talked to him about it. And nobody has replicated it either in 25 years. We're working on that. Could psilocybin really increase the brain metabolism so much? I just don't know. Something might be going on that we just don't understand. Because really, if you gave amphetamine at a high dose to someone, you get these little tiny increases in glucose consumption in the brain. And antidepressants like SSRIs, they produce slight changes. Very few drugs are producing such a big increase. So I was able to pursue this first. Well, the SNF kindly released funding to do this kind of experiment, which is still underway. We're in the final stage of data collection. But meanwhile, because I was too impatient, I went to my friend Mikkel Palner's lab in South Denmark, and he agreed to host a very simple, really a pilot study to test this phenomenon in rats, where we gave rats, pre-treated them with harmine, the MAO inhibitor, DMT, the psychoactive compound, separately or in conjunction, and then injected fluorodeoxyglucose, zipped them into the scanner, and took a snapshot of the glucose uptake. And the figure represents here the results, where these are horizontal planes through the brain of rats, averaged. Each of these images is from 6 different rats that have been stacked one on top of each other and averaged. And you see there's a heterogeneous pattern of glucose consumption, sort of higher in the anterior cingulate cortex here. And here's the conditions: vehicle, harmine, DMT, and DMT plus harmine. Spot the difference. Well, there really isn't any. It did nothing to glucose metabolism. We don't know what to make of that. There was no 20% increase. If there had been, we would have seen it. In retrospect, we probably gave a rather low dose of 1 milligram per kilogram. We probably should have gone for 3 milligrams per kilogram. But nothing jumps out at you. Maybe something going on here in the thalamus. In fact, that's kind of what we saw was that there was a slight increase in glucose consumption in the thalamus in the harmine plus DMT group, but very far from significance because the variance of these measures is so high. We would have had to have 20 rats per group in order to detect the effect. That's not going to happen. So we're now proceeding and well underway. I think Clemens has scanned 14 participants, recruited in Zurich, scanned them at baseline with FDG in the human scanner, and after giving them, in a semi-blinded manner, a tablet containing DMT plus harmine at a psychoactive dose. Unfortunately, I don't know the results yet. We're all in suspense. But this is the kind of picture we get these days with modern PET scanners, showing how extensively the brain lights up with fluorodeoxyglucose, even when this person is just sitting there. So we'll be able to evaluate the effects very soon, in a matter of weeks, I think. I'll have an answer to see if it did anything. And we know that it was a psychoactive dose because they were meanwhile interviewing the participants while they're sitting in the scanner, basically, how high are you? And I think we got a 6 or 7 moderately intense experience, which is what we aimed for. We didn't want them tripping balls in the scanner. Anyway, returning back to the rat studies that we did in Denmark, I also, coming from a neurochemistry background, I just wanted to simply measure the concentrations. How am I doing for time? Another 5 minutes or so?
No, you have all the time you need.
Don't say that. You might want to go home. The returning back to the rat studies, which I did in Denmark, I was interested in just simply measuring the concentrations of these substances in the brain of these unfortunate rats. Not the same ones as got the fluorodeoxyglucose. And so we administered 2 different doses of DMT plus harmine, both drugs together or separately sometimes, at a low dose and together at a high dose of 3 milligrams, which is probably directly comparable to the doses sought by humans. would be something like 3 milligrams per kilogram. And then afterwards, after an hour had passed, the rats were killed, and I dissected their brains into bits, and we sent them to, I hope you're okay with that. I feel like I need to justify it. I mean, they're giving this stuff to people, and we still don't know what it does in the brain. So, and sent to Zurich to the forensics department to measure the contents of various biomolecules, including DMT and its metabolite, and serotonin and its metabolite. And it's a complex figure, but if we just look at the DMT, the vehicle alone group, there was really not possible to measure DMT. And I mention this because there is, as we shall see again, a long tradition of thinking that DMT is actually a natural substance in the brain. It is, actually, but it's not very abundant. The concentrations here in nanograms per gram are very low and hardly significantly different than zero. And in the rats that were given DMT and killed an hour later, we still couldn't measure any DMT in the brain. But what we could see was that there was a huge increase in the metabolite of DMT, 3-indole acid. And since it doesn't cross the blood-brain barrier readily, we know that that indole acid was formed in situ in the brain. So in other words, DMT did get into the brain, but it was immediately broken down by MAO in the brain, yielding this metabolite, which is inert and does nothing. Whereas in experiment 1 was the low dose and experiment 2 was the high dose, only in that condition, when you give harmine and DMT, you get really quite substantial concentrations of DMT in the brain. And there's still some leakage, it's still breaking down to 3-IAA, but at the high dose of harmine, 3 milligrams per kilogram, it's pretty much 60% blocked, or 80% blocked in brain. So in effect, we're measuring the IC50 for MAO inhibition. It's kind of a secondary outcome here. But the main point, the main take-home lesson, well, there's two. One is that we can't measure DMT in living brain unless, by this technique, the concentrations of endogenous DMT, I mean naturally formed in the brain, were too low to measure reliably. That if you give DMT by itself, it gets into brain, but it's immediately broken down. And that if you give harmine plus DMT together, you can get quite substantial DMT concentrations in the brain. How high? Well, actually 10 micromolar. And if you have ever worked in a neurochemistry lab, this is a high concentration. I mean, we usually deal in nanomolars quantities. So it's macroscopic amounts of DMT are getting into the brain of the rat in this condition. And surely that's would be enough to exert actions on serotonin receptors, you would think. We did the experiment using an ex vivo technique where the rats were treated with various combinations, harmine, DMT, etc., and then were injected with a serotonin ligand. This is done without a PET scanner, but sort of offline. So the expectation is that if DMT is exerting its effect at serotonin receptors in the brain, then the DMT condition should reduce the binding of the serotonin ligand, yeah? I mean, because the DMT is binding to the receptors, the more it's binding to receptors, the less are available for binding the radioactive ligand. And as you can see, there was no effect. In other words, at 1 milligram per kilogram quantities, we couldn't discern any. Is this 1? Yeah. We also did it with 3 milligrams and still couldn't see any occupancy. Whereas from this beautiful work from Copenhagen, which is kind of the center for molecular imaging and psychedelics, you see the binding of serotonin receptors in the cortex of healthy volunteers at baseline and after psilocybin at a psychoactive dose. It's night and day, there's no question about it. The psilocybin substantially sticks to the serotonin receptors, thereby producing its psychoactive effects. Whereas so far, we haven't been able to establish this with DMT. And it's a mystery. We're trying to proceed further in this, but there's some possibility that DMT binds to intracellular receptors, that it may not be acting like a classical pharmacological substance binding to plasma membrane receptors, such that it just simply didn't seem to stick, but yet it's doing something. Is there some methodological reason why we could not detect occupancy? By occupancy, I mean how many of the serotonin receptors is the DMT sitting on and making, therefore, unavailable for binding with another ligand? Could DMT actually, like perhaps DMT, be acting at some other site? It does bind to all of these different receptors. It's about as non-selective as LSD, but it doesn't bind to any of those receptors with high affinity. So at this point, we just don't know. And since I have a few more minutes, yes, I'm just going to try to wrap up a little bit, which is that returning to this question about whether DMT is an endogenous neurosubstance. Yes, using more sensitive methods than we had, you can detect DMT in the brain. And how might it be acting? If it's as a neurotransmitter, or how might it be acting if you administer it orally as a medication? Well, here's the DMT molecule, dimethyltryptamine. There's several preclinical, rat studies that suggest that DMT actually is sort of a Trojan horse molecule. It gets into the serotonin neurons themselves because it is able to be transported by this site of action of the serotonin-specific antidepressants, the serotonin transporters. And once in the, as indicated here, which could be blocked by citalopram or any SSRI, yeah? And this is all based on in vitro studies. The other arm of its possible action as a kind of a false neurotransmitter masquerading as serotonin is that other lines of evidence suggest that it can get into the vesicles that are present in serotonin neurons, maybe booting out natural serotonin and replacing it. That's a hypothesis. And how would you test that? Well, administer DMT to rats, but pre-treat some of them with blockers of those specific processes here in the red boxes. Give us escitalopram, which is an SSRI, and it should block any uptake occurring into the serotonin terminals. Or in other animals, give this compound, which is like reserpine, and it should block the uptake into vesicles. And so to test that hypothesis, we did this other experiment, which is still underway, where we administered DMT to rats, DMT and harmine, and it's hard to read, but we had different groups here. We had vehicle rats that only got the DMT, rats that got DMT plus the serotonin blocker, uptake blocker, and rats that got the other compound that blocked the vesicles. And what you see, first of all, is that, excuse me, I couldn't read. This is the natural serotonin content of the brain, not the DMT. And the serotonin content in the control animals was ranging from very low in the cerebellum to very high in the thalamus. And by giving these drugs, we altered those concentrations. So we could play around with the fate, the destiny of endogenous serotonin by administering uptake blockers and so on. That's been known for forever. This is like a replication of something from 40 years ago. But now focusing on the exogenous substances, the DMT that we administered, because this figure refers to the serotonin that the brain is naturally producing, and I emphasize the serotonin concentrations vary a lot between the serotonin-rich regions and the serotonin-poor regions. Whereas if you give DMT exogenously, it gets into the brain and bathes the whole brain. Nonetheless, the hypothesis that we were testing was that it wouldn't distribute evenly in the brain, but it would track the serotonin levels. If DMT is tricking the brain into thinking it's serotonin, the DMT concentration should be higher in the serotonin-rich regions and lower in the serotonin-poor regions. And that's exactly what we didn't see. It's basically flat. Across every brain region and across every condition, the DMT levels and its metabolite, hydroxyindoleacetic acid, were practically the same, except maybe in this region where I probably contaminated the samples. So much. I wonder, why is it that all my beautiful theories come out negative? I don't know, but neither citalopram or the other drug that blocks vesicular transport, despite profound effects on the endogenous serotonin, and its metabolite, 5-hydroxyindoleacetic acid, didn't have any effect on DMT uptake and distribution in the brain or its retention. So it was a nice theory, but I think we can reject the hypothesis that serotonin neurons compartmentalize DMT. However, we have some further experiments to conduct to determine things like what is the actual rate of DMT synthesis in the brain. It's normally present at low concentrations, but it could be turning over very quickly. As soon as it's formed, it breaks down. So we're going to do a few other experiments to complete that study. And just why it is important, the enzymatic formation of psychotomimetic, that includes DMT, metabolites from naturally occurring compounds, dated 1961 and authored by Julius Axelrod, who showed way back then that the rat brain contains all of the enzymes necessary to produce DMT from the elements of tryptophan. And here's the guy. I never met him, but he also, as if that weren't enough, he also worked out melatonin synthesis in the pineal gland, and he discovered acetaminophen as an analgesic. And he shared the 1970 Nobel Prize with two other very important researchers for their work on brain dopamine and serotonin. I don't know why he's missing an eye. Don't ask me. So it's an old story and an attractive story, but we at this point really don't know to what extent brain DMT naturally produced is physiologically relevant or not. I suspect not, but I think we need to do a little bit more research to nail that down. I mean, why are the enzymes there if it's not doing anything? That's a rhetorical question. Maybe they're just there, and we don't have an explanation. Generally, enzymes are present if they're doing something important, but not always. So I'm just going to finish with that study, with the neurochemistry study, by doing some further experiments to play around with this. For example, this is probably getting a bit far afield, but I think we can get at how much DMT is being formed in brain by blocking the egress of its metabolite, because the indoleacetic acid is formed by MAO almost immediately, promptly after a particular DMT molecule enters the brain, yielding this metabolite that normally diffuses out of the brain slowly. But you can block that diffusion process with a drug called probenecid. And so I think we'll do a few more rat studies to see if we can get at some numbers reflecting the turnover of DMT in living brain, the naturally produced DMT. And so this is my extended working group, including researchers at Fribourg and mostly at Munich, in Zurich, and Mikkel Palner and my student Clemens Egger. And so I traditionally conclude my little talks with a little bit of pretentious poetry, which is a line from T.S. Eliot, but dating from 1917. As if a magic lantern threw the nerves in patterns on a screen, would it have been worthwhile if one should say that is not it at all, that is not what I meant at all. PET is the magic lantern. Thank you very much for your attention.
Thank you very much, Prof, for this very interesting talk. I think we have time for maybe 15 minutes of questions.
Sure.
Then we can have a drink together and
Yes.
we can proceed.
Don't be.
We don't have a mic for speaking, I think, so just speak loud.
Yeah, thank you for a nice talk. That you didn't see any displays of ketanserin by DMT or MODA, DMT. Could
Yeah.
it be partially because ketanserin has a much higher affinity for the 5-HT2 receptors? That it's not like.
Well, it's the other way around. We thought that the ketanserin should displace the alkaloids, and couldn't see that either with MeO-DMT in PET or in the rats with the converse thing, which is that the DMT failed to displace the ketanserin. Sorry, it was two converse experiments, yeah. Well, affinity isn't really as important as you think. It's the amount that you give. So even a low affinity binding site given at high doses will saturate the receptors. And conversely, at the dose of DMT that we got in the brain, 10 micromolar versus an affinity of ketanserin 1 nanomolar, basically there was a 10,000-fold excess of the DMT over the ketanserin with respect to the binding site. So it really should have displaced it. Something else is going on. But it's a good point, yeah. I guess I'm taking... Yeah. Sorry, go ahead. Yeah.
So, we know that other classical psychedelics like LSD or psilocybin have tolerance effects. If you take it, you need to take more the next day, etc. However, as far as I know, it's not the case with DMT.
That's correct, yes.
So, do you think the absence of a tolerance build-up is related
to this some kind of anomalous binding property of DMT? Yes, I think so. I think, put another way, we don't really understand yet how DMT works on the brain. It behaves differently to the other psychedelics. Specifically with respect to the tolerance, which seems to be non-existent. Has anyone actually systematically looked at that, or is that anecdote?
I think there is some studies, but there is also a lot of anecdotal data.
Yeah. But that's just a distinct mechanism that we don't understand yet.
Maybe two question. What is the exact use, if we know it, of the 5H2A receptor in the brain, and the other one, DMT is present in plants. Plants don't have any brain.
You asked me what a neurotransmitter receptor actually does. It's very hard to answer that kind of question. I mean, it activates second messenger systems and alters the responsiveness of the neuron that bears the receptor to other stimuli. But serotonin agonism has some effects on mood and cognition, but it's pretty hard to actually nail that down to say what it actually does. Very recently, people did a study in non-depressed individuals who were given citalopram, an SSRI, and it potentiated the activation of their amygdala in response to visual cues. So it's somehow setting the gain of responsiveness of the nervous system to other stimuli. I'm afraid I can't give you a clearer answer than that, because yes, exactly, yeah. So it was just an interesting result that SSRIs seem given to normal people have some kind of potentiation of emotional responsiveness, as distinct from whatever effects they may have in depressed people. As for the second point of the question was, why do plants produce these compounds, or toads for that matter? There's this caution against using teleological arguments, that it's serving a purpose. But I believe that it has to do with insect predation. Oftentimes, insect predation. So, for example, DMT, the mimosa plant, which is where they used to source DMT in Zurich from this, not just the plant, but the roots, the root bark of the mimosa has almost 1 or 2% DMT by dry weight. So it's a very good source for extracting DMT. And who eats the roots of the mimosa? Well, worms, nematodes, basically. And so these substances, first of all, they're alkali, so they're bases. So if you tasted them, they would be bitter. So in fact, with the DMT harming tablets that they developed in Zurich, they try to mask the bitter taste with flavorants. But it's probably also directly affecting the nervous system of predatory insects. I mean, that's why people think that tobacco plant produces nicotine. It's an insecticide. And in fact, tobacco plants apparently respond to insect predation by increasing their nicotine levels. The plants they don't have a brain, but they can tell if someone's chewing on them, let's say. Yeah.
Another thing I know is like 5-MeO DMT is known to be lethal to dogs, but it's not for humans, or at least not in the same.
It's lethal for dogs?
Yes. So, it
Well,
is known
this is news to me.
yeah, it is known to be lethal for dogs and possibly other mammals, and that brings the question, because it reacts differently for humans and dogs, could it be that it also reacts differently for rats and humans? Like, can we really predict the effect it will have on humans based on what we get?
Or rat studies, for that matter. Yeah, exactly. We don't know. I mean, that's why one of the reasons we're doing the human study is to try to see what its effects are in the human brain. I was unaware that MeO-DMT was toxic to dogs, but so is chocolate. It's because of the theophylline, and I think the explanation there is that they can't metabolize it, right? So it has a hyper...
So I don't know the exact reasons, but yeah.
Yeah. Well, yeah, that's it's animals are not all alike. Species differ with respect to their responses to drugs. Sure. Yes.
So you explain that the brain has DMT and it can produce DMT in special occasions. So can you remind me if it's moment like close to the death?
Oh, yeah. Right. That idea was floated around, but I'm afraid I don't believe it. I mean, first of all, we had to give a mass 50 milligram per kilogram, 50 milligram doses to people, or 100 milligrams of DMT to produce psychotropic effects, right? And how much DMT could there be, is there in the pineal gland? It's microgram quantities. On the other hand, the pineal gland produces melatonin at physiologically relevant concentrations. But I don't think that the DMT potentially released by the pineal gland could be psychoactive. It might be doing other things. You know, it might be binding to some other receptor in the blood, in the circulatory system. We just don't know. But I have to discourage thinking that the pineal gland has enough DMT to produce any kind of experience. Even if it were all released at once, it's just too little.
It's something that's also produced in the lungs.
I'm sorry?
I think it's also produced in the lungs.
Yeah, the highest, the highest level of the enzyme, indole N-methyltransferase, is in the lung. Yes, so that itself suggests that there might be a cardio, or a vascular effect, that it might be altering tone of the pulmonary vasculature. That could be, yeah. And it's present in some brain structures. The enzymes are present in some brain structures, including the choroid plexus, I think. So it might have something to do with ion balance in the brain, but the psychoactive component is apparently much harder to establish. Yeah. Yes?
Thank you for that, and I'm coming from a different field. I'm not a scientist, I'm a literature teacher. I can tell you about T.S. Eliot some more, but my question's, so I read Michael Pollan on this, right? I don't know about the details. I can't ask you a question about neurotransmitters, but I have a question for you. I wonder if you'd be willing to take a shot at it.
Yeah.
I'm interested in, so one of the things that distinguishes psychedelics from other kinds of intoxication is the noetic quality. The quality that people experience of it being extremely real, extremely meaningful. People, these studies talk about the most important spiritual experience they've had, as opposed to when you get really, really fucked up on alcohol. The next day you were really fucked up on alcohol, and you're not going to, take seriously the things that you said or thought or whatever. So there's like a difference there. It seems to be.
Yeah.
How would you explain that noetic? I mean, as a scientist, coming at this from neurotransmitters and chemicals, how would you explain that extremely meaningful, lasting experience that it gives?
Well, this is recalling a conversation that, I'm sorry, your name? Pierre-Louis and I had before the talk. which is, the compelling nature of the experience. And we were talking about machine elves and how I met someone, some illustrious psychonaut at a conference, and asked him, are they real or not? And he said he didn't care. And maybe it's something about the experience that this kind of critical reflexes are suspended. Maybe it is an essential aspect of the experience. But why would that be different? Yes, in fact, I had a bit to drink after the election, and it didn't change the results. What would make it different Are you sure? Couldn't there be some psychonauts who would have this profound experience and say, enough of this tourist stuff? I don't know. I don't have very much or practically no experience in this myself, so I don't know. Is it possible to reject the veracity of the experience while you're under the influence? I don't know. Anyone tell me? Because it could be, I can imagine it going both ways. It might be that, as you say, this noetic aspect is wired into the experience such that you cannot separate it.
Maybe not rejected, but adopted, yes.
No. In medicine, in psychedelic medicine, we know that there is always a dissociative effect, which is even a criterion for psychonautism. You always have a foot in the reality and a foot in the fantasy.
Even at the highest doses involving ego dissolution?
You know that you are on a trip. I can tell you.
Okay. You know that you're on a trip, and yet you generally don't discount the validity of the experience.
That's just reported by a lot of...
Yeah, sure. No, I've heard this, exactly.
It's not that, everything you experience is real. It's more like the deeper meaning of that experience is existentially, I don't know.
But is that, sorry,
Yeah.
is the reverse also reported, or are we just reporting the positive trends, right? Like these, because I've heard somebody rejecting having an experience and saying it was not their fault or it was not. So what is being reported?
As a psychiatrist, I have to say that it is an imaginary reality, but it is a reality. It is a psychic, fantasic reality. When you are in a delirium, you are in something like it, but you don't criticize the state. You just involve quite 100% into your psychic modification. When you are in a dissociative state with psychedelics, you have this foot in the reality and one foot in the imagination. But this is an object. This is a real object. It is a psychical object. It is real for you when you live it. It's a kind of reality for your brain. For your mind. For your soul. Because I'm not a neurochemist. I don't know the details of your research. But I know psychiatry, psychoanalysis, and spiritual medicine. And the modification of brain, mind, and soul have to be equilibrated. And this is why you have your question about, is it real?
Well,
Of course it's real.
The people have long since, I guess, abandoned the idea that psychedelics are any model for psychosis, but then apply this analysis to psychotic experience, an actual psychotic episode. People can't tell the difference, I guess.
No, they can't.
So,
But it's acute state.
in the acute state, yes, yeah, and then when they respond or remit, then they. Okay, so then we're getting at some kind of distinction between the psychotic state of being able to, not being able to separate and have one foot in both camps, whereas the psychedelic experience is, let's say, unless there's underlying psychosis. I mean, how dangerous do you think psychedelics are for people who are psychotic?
No, it's a contraindication.
It's a contraindication, but how dangerous is it? That's what I'd like to know.
Yeah.
Very dangerous?
For a psychotic person,
Yes.
I mean for a schizophrenic.
Yeah.
Yeah, it's a contraindication in the Swiss psychedelic.
It is, but I mean, it sort of begs the question, to me, how bad would it be? I mean, would it necessarily produce a relapse or an exacerbation, do you think?
No. But you could be in an acute psychotic state, which is over the border.
Yeah.
More acute than with the normal illness, I would say. The super schizophrenic episode.
Well, amphetamine will do that also to a psychotic. I really, it's outside of my not comfort zone, but I don't really have the terms to frame this kind of question. But I agree completely that it's a fundamental thing that can't really be investigated by measuring chemicals. It's a phenomenological thing.
I asked it because I thought maybe you could, in the sense that you could say, well, this affects the part of the brain that has to do with ego and identity, and so therefore.
Oh, okay. Well, there's some. Yeah, I mean, the people from Imperial College, David Eretzer and David Nutt and others are doing all this very interesting work. Robin Carhart Harris, now in San Francisco, where they used fMRI to look at the functional coupling of different brain regions, and the psychedelic experience. I think they were working with psilocybin. Is somehow, this isn't my field, but the segregation of brain circuits that kind of naturally fall into coherent patterns seems to disappear, and so it becomes sort of maybe ego dissolution is the experience related to the degradation of the nodal organization of the brain, so that every part of the brain is talking to every other part at the same time, instead of, which may not be a good precondition for escaping from a bear if it's attacking you, you see. Maybe we need to have these nodes in sort of focused organization of the brain between different brain regions in order to live in the world. So, yeah, I mean, I think there is a considerable amount of research in that direction. And specifically, yeah, they're finding, correct me if I'm wrong, that the psychedelic experiences maps to a kind of a homogenization of brain function, so that the separate, the different functional domains of the cortex lose their independence.
The tractography is showing that.
And the tractography, yeah, that's exactly, yeah. And that's best studied using, PET is not the right technique for studying that, because with PET you have to record a certain state that prevails for 60 minutes or so in order for chemicals to bind and so on. But with fMRI they can look at the second or minute scale of the functional state of the brain. Yeah. So here's the thought experiment. If you had one of these fMRI scanners and could put people in it, what could you do? If you could have them press a button to, in effect, gate the recording when they have a certain kind of experience, there will be necessarily a delay because it takes a second to react to anything. But that might be a way of getting at that. We were talking about machine elves. What if you press the button when you meet the machine elf? Just as a trivial example, would that show that? Machine elves are a cool idea, I guess, because it sounds prima facie like a fragmentation of the ego into a kind of subordinate, or they seem independent. That's the core question. Are they actually autonomous creatures living within us, or are they actually, how could we put it? What's the word for the three-dimensional photographs? Holographic representation, in effect, so that the ego, as it's normally constituted, is an assemblage. I mean, when I was a teenager, I was very much influenced by Jung, and this is kind of like mapping to archetype theory. That there could be this sort of hierarchical organization of the brain, and due to this particular type of drug, by whatever means, it somehow allows the little components of your brain to become semi-autonomous rather than integrated. Does that make sense? Yeah. How that is obtained, why serotonin would be the thing to do that, I have no idea. Yeah.
I mean, we know the brain is capable of running multiple egos in case of DID and stuff like that. So, I could wonder, like, maybe those components are always autonomous within the mind, but we are not consciously aware of them. And psychedelics are known to be kind of boundary dissolution agents, so maybe that will give you a view on those autonomous process, I guess.
Well, we habitually rely upon subroutines. Like when I pick up my glasses and fidget with them, it's not conscious, right? It's a routine. So a lot of what we do, in fact, like finding your way home from the bus stop, is not conscious. It's
most of the compute we do is not conscious.
Yeah. Sorry, you wanted to ask something?
Yeah, I don't know if you have any input, but I'm not a professional too, and I'm only trying to relate what I learned through meditation to my own experience. And I could have said that DMT was specific and quite different from the other psychedelics. And it looked like, in my perception, that the other psychedelics are like making connection to memories and exhausting senses, but there is always kind of link with your normal life. And at the time you make the experience of DMT, for sure, I don't know, and you have not given the answer, what specific receptor is active or what specific part of the brain is involved, but you reach a place where the experience is really different. And in my experience, what I've said to people after those kind of experiences, and it's for sure false somehow because it's me, but I'm saying it's not me. This imaginary I've seen, the experience I've seen look like to be too far from my life, too far from my normal memories. It looked like that there is kind of doors or specific part of the brain that is involved that make me in touch with something that is on a different level. So
wasn't that a Beckett play, I, Not I? I, Not I? Is it Beckett? Yeah, so simultaneously having the sense of the ego, but this dissociation that it's part of you, but it's functionally or perceptually it's completely different. Is that what you're saying?
But I'm still in the kind of the situation that was explained. I have a food on each side, but the food that is, in living some kind of new world, imaginary, is really something I cannot explain to myself. It's not a distortion of the normal reality. It's not memory that are getting back that were hidden. It's really something. What the fuck? Where is it coming from?
Yeah. Well, that's. It's much easier to measure chemicals than to answer these questions. I think that. It's like of the class of religious experiences, right? And to return to this question, are the machine elves, if you should be so fortunate to encounter them, are they smarter than you or are they dumber than you? I suspect they're considerably dumber, but maybe specialized. It's sort of something illogical about having a component of your psyche that's intellectual capacity exceeds your own. Could that be? It seems like inverting the pyramid somehow, but and likewise, yeah, with religious experiences or mystical experiences, people have the same same narrative that you can't tell whether it's real or not, and the veracity is compelling. Yeah. So there's this class of noetic experiences that can be obtained without drugs, also. Yeah. I think that's probably it for questions. Thank you so much.